Hey folks, and welcome to the first edition of AP Biology Zog Science. This is going to be your first video that you're going to watch this year, and we're just kind of going to go over a little bit of an overview of biology and talk a little bit about some of the statistical tests that we're going to be using so that way you're ready for the rest of the school year. So, let's see. All right, there we go. Where do we begin when we talk about biology? Well, the fundamental principle of biology is evolution, and this is an idea that is somewhat recent. Probably in the last 10 to 20 years have we really, as biologists, come to see evolution as kind of the fundamental principle of biology. And evolution is what explains the breadth of diversity seen in the natural world, and it's the unifying theme that kind of ties it all together. Biology is the study of the diversity that we do see in the world, and there are basically 5 major themes that kind of run through it. So when we take a look at biology, we take a look at things from a reductionist point of view a lot of times. And a reductionist point of view is where we are looking at individual components of a complex system in order to figure out what that individual function is. So one way that we can talk about that is with our cells, right? Cells are made up of organelles. Each organelle has a specific function. That specific function, right, we would only really be able to determine by looking at it by itself. However, when we put all those organelles together, now it is able to have the full functionality of a cell and emergent properties show, right? A cell can do things that the individual organelles cannot do, right? And that's kind of the idea of us taking a look at systems biology, where we're actually looking at the entirety of all of the things put together and seeing how they interrelate with one another. So as we go through each of our different topics, we're going to take a reductionist perspective, where we're going to look at the single components, and then we're also going to take a systems perspective, where we look at all of them put together and see what is different and what is new. One of the things that's going to be a common theme kind of running through this is the idea of structure and function. The structure of an organism basically helps to form its function, and the function helps to inform the structure of the organism. We see that with varieties of different four-legged creatures, right? Whether that is the flipper of like whales or wings of birds, bats, forelimbs, right, arms, right, all those things have specific structures because they have specific functions, and their function helps again to inform their structure. We also see this when we talk about hemoglobin, right? Normal hemoglobin has a specific shape. However, when you change the underlying structure, such as when someone has sickle cell anemia, that then changes the function slightly of the hemoglobin, and it can impair its function as well. So structure and function is going to go along with this idea of the reductionism in systems biology in terms of something we're going to see a lot of throughout the year. Cells are, again, the basic unit of structure and function for living things, and all cells share characteristics such as a cell membrane, but we have those split up into 2 main categories: your eukaryotic cells and your prokaryotic cells. And we will spend a lot more time talking about cells individually. We've got a whole unit on that. It's unit number 2, but it is the basic unit of structure and function, and so that's why we kind of need to talk about it and just make sure that we've got that down. All right, theme number 2, processes involve the expression and transmission of genetic information. All right, remember, DNA is the fundamental information-carrying molecule within cells. All right, an interesting question is, where did that initial information come from? We'll talk about that a little bit more when we get to our evolution unit. But DNA contains discrete packets of information. Those packets of information are called genes, and they are the units of inheritance. So we inherit genes from our parents, one set from our mother, one set from our father. We'll talk about all the mechanisms involved in that. DNA's function is a direct result of its structure. DNA is made up of 4 nucleic acids, right, that can alternate, 4 nucleotides that can alternate in order, and its order defines its function. If you change the order, the function changes. Information from DNA is turned into traits through a multi-step process called gene expression. We'll spend a lot of time talking about gene expression when we get to our molecular genetics unit. Theme number 3, life requires the transformation of energy and matter. Transformation means we're switching back and forth from one to the other, right? All cellular activities are basically to do work, right? And work requires energy. Those of you who might have taken physics. And so organisms have to get energy from their surroundings, and they also have to get matter from their surroundings, okay? Organisms obtain energy from a variety of different ways, right? They can either consume it from other organisms, they can produce it from the sun, and then most of that energy is lost as heat. The entropy increases. Those of you taking chemistry, which should be all of you, that's the second law of thermodynamics, right? Entropy is always increasing in a system. We will spend more time specifically on that. Energy flows through an ecosystem, so energy comes from the sun and it moves through. All right, theme number 4, organisms interact with other organisms in the physical environment. So, there's lots of different types of interactions. That's basically going to be ecology, right? Predator-prey, mutualism, commensalism, all that, those types of things. But they don't always just interact with other organisms, right? They also interact with them in the physical environment. So where you are in that actual physical environment is very important, right? Those are our abiotic factors, and we'll spend time talking about that as well. And our final theme, this is the one that we kind of alluded to at the beginning of this video, right? Evolution accounts for the unity and diversity of life. So biologists have identified about 1.8 million extant species. Those are species that are alive. There are probably a lot more out there, but it takes time to identify all of them. And this wide diversity of life is explained by the theory of evolution, right? The idea that we came from a single common ancestor and that we have been modified since then. And that makes sense because there's a lot of unity within the diversity of life, right? So we've got 3 different domains of life: bacteria, archaea, eukarya. We'll talk about those kind of as we move through the course. But despite there being all these different types of organisms, right? You've got bacteria, you've got humans, you've got dinosaurs, you've got reptiles, birds, right? There is unity in life in that there is a shared universal genetic language, DNA. DNA is found in all organisms, and then there is also lots of molecular similarities. So all organisms use ATP. Why would all organisms and all life use DNA and ATP? Well, it makes sense that they would have all evolved from a single common ancestor. So this is why the theory of evolution is such an important theme that ties everything else together. So how do we as scientists do things, right? Well, at the beginning of every science course, you've probably done this, right? You go through the scientific method. We're just going to do a quick, brief synopsis, right? You have some sort of observation. You see something. You ask a question like, what is going on? You come up with a hypothesis about why you think that is happening. You make a prediction as to what is going on. What do you think will happen? You then run your experiment, and then you get your result, right? And this is basically the basic idea of science. We're going to do a lot of labs in this class, lots of inquiry-based labs where we will kind of go through a guided setup where I will take you through. You'll get to see a result, and then I'm going to say design your own experiment. And so you're going to do a lot of experimentation in this class. When we make observations in an experiment, there are kind of 2 different ways we can talk about things. We can talk about qualitative data, or we can talk about quantitative data. Qualitative data is where we are describing things in such a way that does not use numbers. Okay, so we're talking about the look of something, right? We're describing it using words. When we talk about quantitative data, we are talking about measurements recorded in tables, graphs, that kind of thing, where you do statistical analysis. Okay, you are encouraged to do both, right? You should be taking both qualitative and quantitative data because both of them are powerful in different ways. Now, when we actually are developing our hypothesis, right, we are going to use our, general idea of the if, then, because statement. Now, you do not have to necessarily do it in that specific order, right? The main thing is the general idea, right? The idea is that we are making a rational and informed opinion based on our guess as to what we think is going to happen, right? So when we write a hypothesis, we're saying, okay, I've observed this phenomenon, right? I've done some background research, okay? And I believe that the reason why we are seeing this phenomenon is because of this, and this is why, right? So that's the basic kind of if, then, because format, right? If something is happening, I observe this happening, then this will happen. What do I think why that is happening, and because, all right? So that's kind of the general idea. Very importantly, hypotheses must be falsifiable. Okay, you have to be able to say that actually my hypothesis is wrong, and we actually learn a lot when our hypotheses are wrong, right? Because we eliminate answers, and a lot of science is about not necessarily confirming a hypothesis, but rather refuting a hypothesis and then saying, okay, if that's not the answer, then what about this? And then we continue to go through until we actually come up with the correct answer. Now, when we talk about something being logical, right, there are a couple different ways we can talk about things. We can talk about inductive reasoning, or we can talk about deductive reasoning. Inductive reasoning, we're going to use a large number of specific observations in order to derive larger generalizations, right? So when we talk about this is the idea of we have lots of data, lots of experimentation, and then we derive kind of larger generalizations. So we say we have all these specific instances, all these specific instances are true, therefore this larger principle must be true. Kind of the opposite of that is deductive reasoning, where we are going to talk about some generalizations, right? So, I know the law of gravity, right, that all things are going to fall down. Therefore, if I hold this bag up and I drop it, it's going to fall, right? That's deductive reasoning. So we can work both ways. Either way, both of those are logical reasoning and are totally valid. All right, now, when you do experiments, okay, it is not just enough to get data, right, and then say, okay, my number here is larger than my number over here, right? Because what we don't know is whether or not that is actually what we call statistically significant, okay? When we're saying something is statistically significant, we are saying that the chance that our results are what they are is not due to random chance, okay? Or it's that the idea that it could be due to random chance, but that is a very low percentage. And what we generally are going to use is what we call a 95% confidence, right? We're going to say we are 95% confident that our results are not due to random chance. And that's when something is statistically significant, okay? And we will spend a lot of time going over this, lots of practice throughout the year, okay? Now, there are three main tests that we are going to be using in this class. We're going to use a chi-square test, which will be on the AP exam, a t-test, and an ANOVA. The t-test and the ANOVA will not be on the AP test. However, the t-test and the ANOVA are the two most widely used tests in the world by biologists, and therefore I want you all to be able to use them as well. And so you'll use them a lot when you do experiments and things like that. So the first test is the chi-square test, and the basic idea is that we're going to look at the difference between an expected value and what we actually got during an experiment. So this is particularly useful in genetic studies, right? Because in a genetic study, let's say that we're trying to look and see, is the distribution of males and females in a population within expected values, right? Well, our expected values that we've got 50% males, 50% females, and so then what we can do is we can compare our expected numbers to our observed numbers and determine whether or not that is statistically significant or not. You should be able to do this test by hand. Okay, because you're going to do it on the AP test with only a scientific calculator where you're going to actually have to do the equation. And again, we'll spend lots of time practicing this, so you'll be very comfortable with it by the time you get to the AP test. All right, the t-test. Again, this is one of the most widely used statistical tests in science. It compares the mean of two sets of data to each other to determine whether or not they are statistically different. Okay, so a mean is an average, right? So let's say we're taking data of tree heights in two different locations, right? And we want to see whether or not those tree heights are statistically significantly different from one another. What we do is we find the means, and then we determine how many standard deviations each of the means are from one another. And if they are more than 3 standard deviations, then we know that it is statistically significant, okay? Now, you could do this by hand, however, that is not something I will require you to do. There are online resources. You can do tests in a variety of different ways. So don't necessarily worry about having to do this by hand. All right, the last statistical test is the ANOVA, and the ANOVA is basically the same thing as a t-test. However, it is done with 3 sets of data. All right. Now, because we're looking at 3 sets of data, it doesn't tell us which one is significantly different from the others, right? That's when you actually have to look at your data and see which one is farther away from the rest, okay? Again, this is using a software, using a calculator type of test, especially because this one is a little more complicated than the t-test. All right, steps for using statistical tests, all right? Number 1, you're going to choose a test. You got to think about your data and what you're trying to determine. Are you comparing what you expect to something that you've observed? You're going to use a chi-square. Are you comparing means? You're going to use a t-test or an ANOVA. Number 2, you're going to state the null hypothesis. Okay, this is the most important part because this is also one of the harder parts. Okay, a null hypothesis is not your hypothesis for what you think is going to happen in the experiment. Okay, this is completely different. A null hypothesis states that there is no statistically significant data or difference between the 2 sets of data, right? So what you're saying is that the means are not different. Whatever your data it is most likely due to random chance, okay? So That is a very important part, and writing a null hypothesis is an art unto itself, and we will go over that. Okay. Then you've got to actually obtain results, right? You got to do your experiment. Whenever you're doing an experiment, you need at least 10 sets of data, right? That's your n number. N has got to be equal or greater than or equal to 10. Finally, you're going to present the data. So you present the raw data in a neat table. So you're always going to have raw data in a table. You're going to calculate descriptive statistics such as mean and standard deviation. You should be able to calculate mean and standard deviation by hand. You don't always have to, but you should be able to. Number 5, you'll carry out the test. So you properly use the software, conduct the statistical test, whether that's chi-square, t-test, or ANOVA. And then you're going to make a conclusion. You're going to state whether you accept or reject the null hypothesis. And be sure talk about whether or not there is a statistically significant difference. Finally, this is very important. You never actually prove a null hypothesis, we just accept it. Okay, so remember the null hypothesis is saying that the data could be due to random chance, right? But we don't know necessarily if it was. So we just accept the fact that it probably was due to random chance, given the data that we have, right? And we don't say that we accept the null hypothesis. We never say that we prove the null hypothesis. We just say that we accept it. All right? Great. Well, thanks for watching, and I'll see you guys next time.